Hello. Today our topic is the calculations of total plate counts in the microbiological samples. Which formula we will use? This one is the formula that will be described, that will be recommended in ISO 7218. This one is the colony counter, which have different modes. It may be manually, it has an automatic mode. This formula apply for total aerobic plate count, for all kinds of counting when we go use for solid media, for our counting, for total entire bacteria count, total E. coli, total coliform, all bacteria we go for counting will go general. This formula applied for in the next part we will discuss in details the part of this formula, what will be the actual mean of this formula. As we already discussed, this one is a formula for total plate counts and is a number of colonies per ml or per gram of the product. Summation C. Summation C is the sum of all the colonies in all plates, especially we will use two dilution, but maybe we will use two plates, or we will use one plates in each. But in our examples, we used only one plate for dilution, first dilution, that will be the transfer -1, and second dilution one plate also. Thus, the formula will become shortened. Thus, C is the sum of all the colonials on all plates in which we will perform counting, and 1 is the number of plates of the first dilution, and 2 is the number of plates in the second dilution counted. And this is the dilution from which the first count were obtained. This one is a very important thing, which is the volume of the samples added in the plate. So we will discuss it with an example. Suppose we have two dilutions, one plates of each. The first dilution is have counts of 38 colonies. Second dilution has number of colonies, and the number of plates we use is 1, and the volume of sample is also 1. Loss dilution factor transfer -1. This means it's first dilution, first initial suspension. When we will apply, the formula will become shortened due to 1 of number of plates, and this will become the short form of this formula. We will add number of colonies. We also add the volume. This one is the dilution factor. In this way, we will calculate the total number of colonies in our samples in per gram or per ml form. How we will calculate by count by using colony counters. In the next part, we will discuss. This one is a colony counters. Suppose there is a plate, and this one is a plate. We want to count. We place in the part of our colony counters. We will simply press the start buttons and give the counting. This one is the automatic process of a colony counters. If there is any kinds of colony that is skipped, we also add it by manually. They also give the option. This one is the first dilution. In the same way, we also go for the second dilution, and we will go for in the next plates. This one is the second dilution plates, and we will count in the same way. In the same process, we will count. We'll count the total number of colonies by the colony counters, and then this one is the colony total number of counts that performed by this colony counter. And in the same way, we will go for the next our formula for total colony. Thank you so much. Thanks for watching. You can, if you have any questions, you can comment in the chat box. Thank you so much.